Assalamualaikum and hello to Dr. Afiq and Dr. Sufiq. Me Muhammadin Hakim bin Ali will be presenting to you my research proposal, which is the comparison of bioethanol production between wild and online seaweed cultivation of Capapicus alvarezi. So first of all, according to Zuldin and Sapawi 2015, the demand on raw and processed seaweed has increased since 2012 and will be keep increasing in the future. However, this seaweed has harvested from open ocean cultivation and wild habitat. This lead to competition of seaweed utilization as food and other aspects such as biofuels. As an alternative, online seaweed cultivation will decrease the competition on seaweed source. Moreover, sustainable online seaweed cultivation need to be achieved by two aspects, which are the diversity of genera that can be cultivated and the profitable value-added production from the seaweed. Next, majority of the studies that has been done was on the bioethanol production from wild and seaweed farm. Also, several studies been done on the optimization method for bioethanol production. However, insufficient data has been recorded for the online seaweed cultivation performance, especially for bioethanol production. One study has been done by Zuldin et al. 2016, focusing on the growth performance and biochemical composition of Capapicus alvarezi in tank cultivation. Next, the table shows on the various of seaweed species that has been used to study the bioethanol production or the component that related to the bioethanol production, such as biochemical composition and growth rate. The table also includes the various seaweed sources that been used by the researcher, such as open water farm, seaweed residue, tank cultivation, laboratory cultivation, and wild habitat. So for the justification, Capapicus alvarezi will be used in this study due to the extensively being farmed in Malaysia for the carrageenan extraction, make it easily to be accessed and acclimatized. Next, online seaweed cultivation will be performed because it can produce high quality and quantity of the seaweed under controlled condition, where many disadvantages from open seaweed cultivation such as herbivorous activity can be avoided. Last but not least, the study will be done to fulfill the knowledge gap where bioethanol production from online seaweed cultivation is poorly known. So, two main objectives in this study are to study the difference of bioethanol production from online seaweed cultivation and wild seaweed of Capapicus alvarezi. Second is to determine the nutrient and light intensity impact on the bioethanol production from both seaweed sources. Here is the materials that will be used in this study. Move to the methodology. Sampling of wild Capapicus alvarezi will be done at 3 different locations, which are Pangkor Island Perak, Langkawi Island Kedah, and Salaka Island Sabah, based on the previous study that used Capapicus alvarezi as their subject. Wild Capapicus alvarezi will be collected by scuba diving or snorkeling and later will be preserved inside a styrofoam container in cool condition for transporting to UMT. For Capapicus alvarezi from Sabah, the seaweed will be posted using domestic courier under same condition. After that, at UMT, some of the Capapicus will be acclimatized before being cultured inside a tank, and some of them will be proceed for ethanol production. For the online seaweed cultivation, Capapicus alvarezi will be cultivated inside 7 tanks with different nutrient and light intensity according to the table shown. Cultivation will be done for 40 days. For the in situ parameter and water sampling, YSI multiparameter will be used to measure the temperature, salinity, and dissolved oxygen inside the tank, while light meter will be used to measure the light intensity. Meanwhile, for wild habitat, diving pump will be used to measure the water parameter. Water sample also will be taken from both conditions for nutrient analysis using Niskin water sampler. Next is the bioethanol production. Bioethanol production contains 3 major steps, which are pre-treatment, hydrolysis, and fermentation. Pre-treatment will be done by washing and drying the Carpophycus alvarezi to remove any foreign substance and to remove moisture from the biomass. After that, it will be green and sealed for hydrolysis process. Hydrolysis process is to convert any complex sugar into simple sugar. Enzymatic hydrolysis will be used due to the effectiveness in converting compared to the other method, and it will reduce the byproduct production. Cellulose 1.5L and viscozyme L will be used due to the well-known and commercially available. After the hydrolysis process, fermentation process will be done using yeast called Saccharomyces cerevisiae. The fermentation will be done for 72 hours. After 72 hours, ethanol concentration will be measured using gas chromatography with flame ionization detector. Standard curve will be constructed using known ethanol concentration and be compared with the samples. So, for the data analysis, two sample t-tests will be used to determine the significant difference between ethanol production from different sources, and two-way ANOVA will be used to determine the significant difference between nutrient and light intensity on the ethanol production. Two key tests will be followed up to determine which nutrient and light intensity affect the ethanol production. So for the expected outcome, bioethanol production will be high in on-land Carpophycus alvarezi cultivation, and optimal nutrient and light intensity will produce high bioethanol concentration. So this is the Gantt chart. The study will be start on September 2021 and end on August 2022. So next is the reference that been used in this slide. That's all from me. Thank you.